Hi, and welcome to this lecture on environmental adaptation of animals. On this series of molecules and cells in animal physiology, it's something that you should already be a little bit familiar with, like the enzymes. But, however, this video contains a lot of new stuff on enzymes as a capacitor of environmental adaptation. But to summarize, these enzymes are just one type of proteins, so you have about 2000 different enzymes in your body, as well as you have about 2000 different proteins regulating on gene regulation. And all the proteins are formed by an amino acid chain or chains by the instructions coded in genes. And the very basics of enzymes is very simple. So they are factory. They are speeding up the biochemical reaction, like for example cleavage of disaccharide. And this reaction can happen even without the enzyme, but it's very slow. And animals were not very good in waiting, so the speed is insufficient for the living organisms. But still, these enzymes are very important factors to affect that, okay, for example, how we can jump. We have two kinds of amphibians. We have frogs that are good in jumping, and we have toads that are slow. And because all the amphibians are not very good breathers, so there's not enough oxygen available for the leg muscles, especially when they are moving. So they had to rely on the anaerobic metabolism. And this means that, okay, they can cut the glucose with the glycolysis to produce some ATP, and then in the end product they will get pyruvate, and they will form from the pyruvate to lactic acid. And then this lactate dehydrogenase is then in a key role for making the lactate acid. So these enzymes are potentiating the reaction without being used. So the lactate dehydrogenase is using pyruvate or pyruvate acid to form lactic acid. And actually it's doing it, so using at the same time NADH that has been taking protons from the glycolysis reactions, and now it's putting the proton to form lactic acid, so this hydrogen. And then when it's releasing the hydrogen, it's again in NADH form to going back to the glycolysis processes. So it's catalyzing this reaction, and this reaction is moving the protons from NADH. And then this NAD can be recycled in the glycolysis. But then, in the toad, this enzyme is not so effective. So that's why there is an accumulation of pyruvate acid, accumulation of NADH, and there's not enough NAD for the glycolysis anymore. And that limits the ATP production, and then there's no enough energy for the jumping. Well, enzymes can be fast or slow, so they have some kind of speed. And this speed can be tested easily by testing the enzyme activity in presence of different concentration of substrate. Quite often the enzymes are working in hyperbolic kinetics, where adding a little bit more substrate increases the speed of the reaction drastically. Because the enzymes definitely can't work without getting the substrate binding in the enzyme and then start to process it. But when the substrate concentration is very high, then all the enzymes are occupied and it cannot be any more faster. And this is a typical way to have it in hyperbolic, but in some cases you might see that, okay, in some cases the enzyme is working in sigmoidal kinetics, where in low concentration of substrate nothing happens. So in these cases the substrate, there's more than one binding site and these sites are interacting. So it's not working, for example, before there's more than one substrate binded. And what is making it's lowering the activation energy. The substrate and the products are usually stable molecules because even tomorrow there exist these molecules in your body. So there must be an energy barrier to prevent that they are not changing to other molecules. And this energy barrier must be crossed before this reaction can happen. And the activation energy is lower when we have a catalyst. And in these enzyme reactions, we have actually 4 different reactions to happen. First of all, we have enzyme and the substrate. They must find each other to form the enzyme substrate complex. And then this enzyme can work to make the substrate to turn the product. And then the product must leave from the enzyme. And usually the slowest one is to find the substrate and bind it. So that's why we can have different speeds in the reactions. And this activation energy actually can be used in the environmental adaptation. Well, ion transporters, well, they are not very typical enzymes, but they are enzymes anyway. They are doing some kind of conformational change and then binding the ion and then making conformational change and then releasing the ion on the opposite direction of the membrane. And this activation energy can be measured by testing this reaction in different temperatures. From that you can easily calculate the activation energy directly. And in mammalian sodium calcium exchanger, it's losing the activity in cold, but in trout it maintains the ion transportation. And that means that in the right hand side, the trout sodium calcium exchanger, this ion transportation, it has lower activation energy. So although the activation energy sounds very physics, it has some consequences in the animal physiology and in environmental adaptation. Well, how then some reactions can be fast or slow? Usually it's affecting on the substrate affinity. So in enzymes, they can have the same maximum speed, but if the affinity is different, then usually when we have a very small amount of substrate available there, then increasing a little bit if we have a high affinity enzyme, it will be much faster than these low affinities. And if they have these hyperbolic kinetics, it can be very easily tested by using different enzyme concentrations to see that where this speed is half maximum. And then we can compare different enzymes or enzyme types easily. And even these kind of consequences in animal physiology, like in transportation of oxygen through the placenta. Well, hemoglobin, it's not a very good example of enzyme, but it binds oxygen and then it releases the oxygen. And it binds several oxygen molecules, and they are interacting with each other, so it's a sigmoidal kinetics. And you can see that in very low oxygen pressure, there is no saturation at all. But then when the oxygen concentration is increasing, then the saturation is increasing drastically. But the hemoglobin is actually again formed by different globulins, and in adult it's on alpha and beta subunit, or alpha and beta globulins. And then the fetal hemoglobin is formed by alpha and gamma globulin, but this gamma globulin is binding oxygen much higher efficiently. So it has much higher binding affinity, and therefore this maternal red cell is losing the oxygen. So the fetal red cell is stealing the oxygen when they are near each other. Well, actually all the reactions are reversible, and that's why we have a mass action that is affecting. So even this lactate dehydrogenase, lactate can be modulated to form pyruvate, or the pyruvate can be modulated to form lactate. And then depending on the amount of these reactants, pyruvate, NADH, lactate and NAD, the reaction can either produce lactate or pyruvate. And then we can have also equilibrium where the reaction speed in both directions is similar. But in living animals, NAD is used in the cell, and sometimes the lactate can be also secreted. So then that actually means that, okay, the reaction is actually going only in one direction. Okay, how then we can have easy way to have a enzyme affinity, enzyme effectivity different, is that actually we have more than one enzyme of the type. So we have different isoforms, or in enzyme we have calling them isozymes. Like lactate dehydrogenase, actually we have several different types in your body. It's composed by 4 subunits, so it's a tetramer, and usually these subunits are muscle type or heart type. So in your heart or your brains, you have an isoform with 4 heart type subunit. And then in the liver or striated muscle, you have isoform with 4 muscle type subunit. But then we have also these combinations. So we have in lungs, we have isoform with 2 heart type, 2 muscle type isoform. So it depends in different tissues, and the easiest way to make the adaptation is that actually you could start using another type of isoform in certain cells. So when we have different types of these of enzymes, they of course means that, okay, they must have some kind of differences. And one of the things that I was is not in the table at all. Actually, we have in the testes we have a completely different isoform without subunits of heart type or the muscle type. So there are some other types also. So they can have differences, and one of the differences is that in this muscle type it has higher pyruvate affinity, and that's why it's a little bit more efficient to produce lactate. Okay, we can regulate the enzymes either by changing that, okay, we have different isoforms, so if we regulate, increase the amount of enzyme, or change from one isotype to another isotype, but then we have also this protein level regulation. So we can have 2 ways. We have slow way and fast way. We have fast way is this allosteric regulation. The slow way, so to affect on the gene regulation, it takes always a lot of time for the protein synthesis and etc. But still it can be used for, especially for rarely faced challenges, like cytochrome P450. It's found in liver, kidneys, gastrointestinal tract, and they detoxify the foreign compounds. And even a single exposed to inducing agents, anesthetic drugs, pollutants, hypoxia, increase the synthesis of certain P450. And then on the next time you face the same challenge, it's detoxifying the foreign compounds very fast. And one of the things that this is for where this is important is because it's affecting by anesthetic drugs. So that's why if you are very often getting anesthesia, so your response for these drugs is changed. So these enzymes can be modulated by gene expression, but quite often they are regulated also by these allosteric modulations. And if we have a biochemical road where there are several different steps, so are we just increasing all the enzymes, activity of all the enzymes on this road? No, because that's not very efficient. The regulation happens on the rate limiting reaction. So that reaction that is the slowest one. Just speeding it up, the slowest one will speed up to completely road to form more the product E. But in some cases, we have a branching metabolic pathways. And these are very interesting, that if we are regulating the enzyme 2 and enzyme 5, it then affects that, okay, are we producing product E or product H? Then we can have from the same reaction route two different products depending on the regulation of these enzymes. So how these enzymes are regulated? Well, actually, they are just another reaction with the mass action principle. So therefore, when we have a reaction from B to C, it can be enhanced rapidly, for example, by regulating the amount of enzyme regulator. So we have an enzyme that is again regulated, for example, M. When we have a lot of M, all the enzymes are have these regulators, and then it can be working properly. But in case of low concentration of M, then these enzymes are not occupied, and then this reaction from B to C is inhibited. And one of the examples of this is using intracellular calcium. So when we are releasing calcium, either through the cell membrane or from intracellular storages, the calcium is then delivering inside the cell and binding on different enzymes and activating them very fast. Okay, let's have an example. For example, glucose. Okay, we are making the anaerobic metabolism. First of all, we have hexokinase. Then we have hexose phosphate isomerase to produce fructose 6-phosphate, and then we have phosphofructokinase. And this reaction is quite much the rate limiting. So this enzyme is regulated by two different factors. First of all, there is citrate, and it's decreasing the catalytic activity. So in case we have a lot of citrate, this glycolysis is inhibited. But then we have also AMP, which is increasing the catalytic activity. So in case we have a lot of AMP, it will enhance the glycolysis to form pyruvate. And so that's why we can have quite a mess. That okay, in case of we have citrate and AMP, then these are competing each other. Okay, will the phosphofructokinase work or not? And that's why these metabolic pathways can be a little bit messy. Well, we also can modulate covalently the enzyme activity. We have phosphorylation and dephosphorylation. Enzyme molecules can have phosphate by phosphorylation. This is made by protein kinases. And then we have enzyme with the phosphate. Then this phosphate can be also removed by a reaction called dephosphorylation, and this is caused by phosphatases. And although in several cases the phosphorylated enzyme is active and dephosphorylated is inactive, that's not always the case. But that's a kind of thumb rule. So let's have an example. Glucose is stored as a glycogen in animal cells. So it's a very long chain of these glucose molecules. And when we need the glucose, we have a glycogen phosphorylase that is cutting glucose from the polymer. Then we have glucose with one phosphate and then a little bit shorter glycogen. And this glycogen phosphorylase is regulated with phosphatase and phosphorylase kinase. So it can be kinase, okay, then it's phosphorylated, it's active, and then it can be downregulated by removing this phosphate. So it's quite simple way to induce then the cleavage of glycogen or limiting the glucose concentration completely. Okay, then when we have these kinases, you all know that okay, they are used for amplification. When we have some activating agent, it's activating one kinase. That is then phosphorylating other things. And it's phosphorylating, for example, other kinases, and so making that okay, adding phosphate on these. And usually it's affecting on more than one. For example, if it's affecting on 4, now we have 4 times higher responses from this activation of single. And then on next level, it's again can affect on different molecules. And if it's affecting on 4 target molecules, then we have 4 times 4, meaning that okay, now we have 16 times larger response for activating this single kinase. So how these enzymes can be compared? One interesting thing nowadays is to use molecular biology. So when we have cloned different enzymes, we can compare their structure. For example, by using the protein comparison. So we know the genetic code. We know that okay, what kind of amino acid they have, and then when we are checking different animals, we can see that okay, certain amino acid exist in certain animals, and then when comparing these, we can make these hypothetical appearing in the evolution to see that okay, in one animal there is this yellow amino acid, but in the other ones there is not. But in these two animals there is this red mutation and etc. And this way we can then compare the whole sequence of practically all the animals. And for example, in lactate dehydrogenase, it's in muscle form, meaning A form, and heart form M, in blue, so in B form. So you can see that the mammalian muscle type there is there in the top, and then the mammalian heart type is there in the lower section. And quite nicely, all these terrestrial all the terrestrial animals, they are divided quite nicely. But then these fish muscle type, they are quite far from these land organisms, terrestrial vertebrates on this muscle type. They are in the middle. And one interesting thing is that okay, actually in Xenopus, so in cleaved frog, there is a yellow dot there in the center of these blue heart types. So Xenopus muscle type is more like the hard type. And that's making very interesting on these molecular tools, for example, these sequence comparisons. And another interesting thing is that, okay, when we are using these molecular techniques, we can test that, okay, what kind of alleles we have in different environments. So we have a killifish that have two different isoforms of this lactate dehydrogenase. And when tested on the east coast of North America, it shifts from one type to another one. So in somewhere, it's 50-50 chance to have B-type or A-type. But then going north, everybody has B-type. Then going south, everybody has A-type. And of course, these animals can travel long distances, but still these allele differences are so huge. So there must be some benefit for the reproduction on the area. Thank you.